All right, we'll get started. So I'm Tom Grant. I'm a staff scientist here at XFEL. I'm involved in a lot of solution scattering studies and work. So I'm going to talk relatively briefly about some of the solution scattering studies that you can do at XFELs. So to start off, we'll just start with some basics of what solution scattering actually is, how it works. So it's based on the same fundamental idea of scattering that crystallography is, that or single molecule imaging is. We start off with essentially the molecular transform, which is the Fourier transform of your electron density function. Okay, and so this is a complex valued function. There's amplitudes and phases associated with it. Where in crystallography, we start with our molecular transform. When we have crystals, what we actually end up doing is sampling that molecular transform according to the crystal lattice. So the crystal packing, how it's arranged in physical space, tells you where these spots and these reflections occur on your detector. And essentially what you're doing is you're sampling that underlying molecular transform. And so this is obviously just a 2D slice from the Ewald sphere through what is in reality a three-dimensional molecular transform. And so you're actually just measuring those intensities, albeit greatly amplified with crystals. But you're just measuring those underlying intensities, but again, we get no phase information from that. With solution scattering, we're starting with the same basic molecular transform, but we have two types of solution scattering that we consider. We have wide-angle scattering, which we call WAX, or wide-angle X-ray scattering, which can go out to the same general resolutions as crystallography at around 2 angstroms, whereas we also have small-angle X-ray scattering, which is really just interested in that very bright spot in the center of your image, which maybe is around 20 angstroms. Now, the big difference with actual experiments in these images is that these images are essentially one orientation, but in reality, we have a solution of molecules, and so they're all tumbling. And so we have spherical averaging, so we end up with this isotropic pattern. This is the basic experimental setup. We have our X-ray source. We have a solution of molecules. They scatter in an isotropic pattern. Because it's isotropic, we can actually do radial averaging in order to get better signal to noise, and we end up with a one-dimensional profile of intensity versus Q, which is essentially related to the scattering angle. Again, we have spherically averaged intensity being recorded, and so you have a huge loss of information because of this, whereas particularly at high resolution, because whereas the actual information content of your molecular transform scales with the volume of reciprocal space, it scales as the cube. Because we have a one-dimensional profile, only scales linearly, and so we, particularly at high resolution, we lose a lot of information. But we can analyze this profile to get information about things like the size and shape of particles in solution. Solution scattering is fundamentally a contrast method. So you have to take into account that it's in a solution of buffer, primarily water generally. And because of that, it's a very low signal. And so we end up with something in the neighborhood of 10% above the background for proteins. It's higher for molecules such as RNA and DNA, which are very high electron density. But it's lower for things like membranes, detergents, and things like that. So one of the ways that we deal with this contrast is we actually subtract the signal from the bulk solvent. And so we collect two experiments. We have one in blue here where we have the protein and the buffer. It also includes things like the capillary scattering, any air scattering, or other contaminants. And we collect just the buffer itself without any protein in it. And you end up subtracting the buffer from the total scattering. You end up with just this scattering of your protein solution. So we can take this profile and we can kind of break it down into a few different regions. Okay, so the very small angles will give us information just about the overall size of the particle, things like the radius of gyration, molecular weight, volume. The mid-range scattering angle will give you information about the shape, if it's elongated, if it's globular or unfolded. And when you get into the more of the wax region, you end up looking at internal fluctuations in the scattering. Okay, so we'll start off talking about Sachs versus wax. So these have been done at synchrotrons for a long time. The first Sachs was done back by André Gagné back in 1939. And so it's been around for quite a while. Sachs is low resolution. It's small angles, as I mentioned. So we're looking at something below 20 angstrom resolution. But the term resolution in Sachs is somewhat ambiguous because while we technically go out to the equivalent Bragg scattering angle of, say, 20 angstrom resolution, like I said, we're losing lots of information content because we don't have that three-dimensional molecular transform. So we're losing a significant amount of information content. So when you collect out to 20 angstrom resolution, you actually have much less information than you would typically get at 20 angstrom resolution. One of the advantages of Sachs, it can be done at very dilute solutions. With synchrotron sources, you can do things down to 1 mg per mil and get very reasonable signals for a range of sizes of protein. Sachs is very good at revealing conformational dynamics on a large scale. So things like, large motions of arms and molecular machines, things like that, are very amenable to small angle X-ray scattering. Additionally, because small angle X-ray scattering contains the lowest angle information, we can do ab initio shape determination. You can get some shape information just from your Sachs pattern by itself. Now, wax data obviously has the advantage that it's high resolution, but as high resolution, it's also much lower signal to noise, and so you need much greater concentrations of solution, something like 100-fold greater than you would get from small angle scattering. Wax can tell you information about local structural changes, little fluctuations in your electron density, but it often requires known structure to interpret. So you can't actually interpret what those fluctuations mean unless you have some kind of structure to tell you how they relate in 3 dimensions. So that's one of the major disadvantages of Sachs. And because wax itself doesn't contain that very low resolution information, you can't do any kind of ab initio shape determination. Okay, so what are some of the things that Sachs can provide? So Sachs can provide information about the radius of gyration, which is essentially just the root mean squared distance of all the atoms to the center of mass of the particle. It can tell you what the maximum particle dimension is. It can tell you information about molecular weight. Not only can it tell you information about the oligomeric state, it can actually tell you how the complex subunits are organized in solution. Very importantly, it can tell you a lot of information about the native flexibility or unfoldedness. It can also visualize those dynamic regions that you may not see in X-ray crystallography, because if you have some kind of dynamic region, it disturbs the order in your crystal, obviously, and so it's essentially a very large B factor, and so you don't get any electron density from those regions. And finally, like I mentioned, you can get a low resolution molecular envelope. So how about wax? So wax is often used for characterizing how a structure changes as a function of some kind of impetus, something like binding to a ligand or some kind of light-induced change. You can identify structural changes if you know what your structure is. You can also look at dynamic and ensemble changes. There are very distinct signatures in wax that you can identify based on the folds of proteins. So you can say, a helix is moving in certain directions because of known helix distances in your carbon alpha chain. One of the things that's often used, that wax is often used for, is screening large ligand libraries. And so this is because wax can give you information about small changes in your structure just by mixing a ligand in. And so you just have to do these experiments. You can do these very quickly as long as you have a high enough concentration, and all you're really looking for is changes. If you see something that's non-zero, you know this is something that is binding to my protein of interest. And so a lot of in pharmaceutical companies will use wax to screen large ligand libraries. It's very useful for validating MD simulations. This is one of the reasons that we know that our water models are actually pretty good in MD simulations, because they match very well with wide-angle X-ray scattering simulations. You can test several different hypotheses. Essentially, you look at a series of different trajectories in your molecular dynamics simulation. You can see which ones actually match your experimental data. Also, you can use this information in modeling programs to refine your modeling. It'll try and steer your structure such that it matches your experimental data. Whereas in either crystallography or single particle imaging, you get a 3-dimensional picture of the electron density function. In Sachs, we don't get that. Not only do we lose phase information because of the inherent nature of detecting intensities and not phases, mathematically, the spherical averaging wipes out any kind of phase information as well. Also, we only have a spherical average of intensity information. And so when you do the Fourier transform of that intensity profile, what you end up with is what's called the pair distribution function. And what this is essentially a histogram of all the interatomic pair distances in your structure. So it tells you information about the shape of your structure just by looking at the Fourier transform of that intensity profile. And this is an example of a few different shapes that you can see, just gross representations. So you see a nice globular spherical shape object will kind of have this Gaussian profile. If you've got something bimodal like this with two different lobes, you get this larger peak at lower distances corresponding to all the short distances within each lobe. And then you've got this peak at larger distances corresponding to the longer distances between the lobes. For something like a rod, you get a left skewed pair distribution function because you've got a lot of short distances and very few long distances. And something that's disc shaped, you'd end up with a more expanded, broader pair distribution function. And so just that simple Fourier transform can give you a lot of information directly about the shape of your particle in solution. One of the nice things about the pair distribution function is you can actually do this at high resolution. This is the way that a lot of people will actually interpret structurally when they have combined Sachs and wax data. They can get high resolution information. You can get very tiny fluctuations and look at things like propensity for different types of folding. You can get even things like beta sheets and alpha helices just from your wax data, or your Sachs and wax data. So this is a nice slide that I borrowed from a recent paper review in Current Opinions of Structural Biology. And it essentially describes some of the dynamics that you have access to at different time resolutions using solution scattering or X-ray crystallography. So on the bottom, we have different time resolutions. At very short femtosecond time resolutions, we're looking at things like bond breaking, isomerization. At picosecond and nanosecond, we're looking at things like side chain rotation and motions of helices. And at longer micro and even millisecond time scales, we're looking at more tertiary and quaternary structural changes. So XFELs, obviously, as you've heard, can get you down into that extraordinarily short time scale region of femtoseconds and picoseconds that tell you a lot about the catalyses going on, what different processes that are going on at extremely short time scales. Synchrotrons can actually get you quite short, down to the even nanoseconds, potentially sub-nanosecond time resolutions. Generally, it's reserved for microsecond, millisecond time resolutions. Sachs can give you information, as I said, about the large-scale changes that are going on. Wax can give you information about higher resolution changes going on, and X-ray crystallography obviously can give you very high resolution information. Okay, so time-resolved scattering essentially has two main approaches. The first is mixing, which John touched on. This has the ability to essentially go to sub-millisecond timescales. I'll get more into it in a second. And is applicable to many systems. That's one of its main advantages. Pump probe, however, even though it requires light-triggered reactions, has the main advantage that it can go down to sub-picosecond timescales, where it's essentially just limited by your triggering mechanism. So in the case of pump probe, your triggering mechanism is that pulse of light, whereas mixing, your triggering mechanism is whatever ligand you're binding to or protein you're binding to, and so it's limited by those diffusion and binding constants. Okay, so one of the things about time-resolved solution scattering is the differences are extraordinarily small usually. Unless you're doing large conformational changes, usually you do, you analyze different scattering profiles. So you're looking at, you can see that the blue and the green overlap almost identically, but there are small changes when you look at the difference. This also gets rid of a lot of experimental errors, such as buffer mismatch. Buffer mismatch is a huge problem experimentally because you need to have exactly the same concentrations of all the different ligands and cofactors in your buffer as you do in your protein, and that's difficult to do even with methods such as dialysis and taking buffer from purification protocols. You can do both stop and continuous flow, which we'll get into in a second, and it's very well established at synchrotrons. Okay, so how we actually do this? So this is from Lois Pollock, who is one of our BioXFEL investigators. And Lois is very well familiar with mixing-based solution scattering in Sachs. So essentially what happens is they create these little microfluidic chambers that have some kind of protein of interest. In this case, this is RNA, or molecule of interest. In this case, this is RNA. And they're flowing in an ion in order to cause the RNA to fold. And so they're actually monitoring, as a function of time, the different scattering profiles by essentially positioning the X-ray beam, or rather positioning the sample chamber, relative to the X-ray beam, such that the delay between the mixing region and the X-ray probe corresponds to some kind of time delay. And so by making this stream longer and longer and longer, positioning your separation between your mixing region and your probe region to larger distances, you end up with longer and longer time delays. And so in this case, this is what we call a Cracky plot. And a Cracky plot that essentially just goes up like this, just continues to increase as a function of Q, is unfolded. And you can see that as we increase in time, we end up with a nice, more Gaussian peak, which shows that it's folded. And so here on the left, they're showing the radius of gyration as a function of time. You can see it's kind of this biphasic situation. And so they were able to actually get information about the folding process of RNA in a millisecond timescale. Okay, so Uwe will talk about this in a lot more detail. I'll just mention it very briefly. So mixing jets are essentially the main way that we do diffusion-based analyses for time-resolved studies. It's a type of continuous flow. It often uses gas focusing. There's one of the main advantages of these mixing jets versus either capillary-based or microfluidic-based mixing is that there's no background scatter from the sample chamber. So you end up with these windowless features. So you end up with just X-rays through solution. Very short time resolution, down into potentially the sub-microsecond range. Sub 250 microsecond range due to their very fast mixing. Okay, so pump probe can be done at either a synchrotron or an XFEL. A liquid jet can be used for the sample delivery. The time delay in this case is slightly different than the time delay in, well, not slightly, it's very different than the time delay for mixing. And essentially what happens is you have some kind of activating pulse, either infrared or optical light, depending on your reaction. And you trigger that pulse such that it precedes the X-ray probe pulse by whatever your time delay that you're desiring. So you can set this up such that the jitter between these at XFELs is on the order of 100 to 200 femtoseconds. And so you can have time resolutions that are very short. One of the major advantages of XFELs, obviously, is there's no radiation damage. So the pulse is so short that it passes through the solution before you get any damage, and it's extraordinarily bright. Each pulse delivers somewhere in the neighborhood of 10 to the 12th photons, and so you get much higher scattering than you would get in a similar pulse structure from synchrotrons. And so your time resolution, you get down into the sub-picosecond regime. So some quick examples. Solution scattering in XFELs was first demonstrated in 2014. They collected high-resolution wide-angle scattering profiles out to about 2.5 angstrom resolution. This study was meant to verify structurally the hypothesized protein quake in light-activated proteins, and they were able to get something like 500 femtosecond time resolution. So the main question that they were asking was, how can light-dependent or light-absorbing proteins, things that absorb photons that have energies that are approximately equal to the folding energy or even greater than the folding energy of proteins, actually, how can a protein stay folded? There's so much heat generated that it should just essentially unfold the protein. And so what had been hypothesized is that there's something called a protein quake that actually dissipates energy from that central catalytic region in a essentially just what you would think, like an earthquake-like mechanism that spreads out that energy on an ultra-fast time scale before the heating from the protein actually causes it to unfold. So these were their results. You can see on the left here, they're different scattering profiles. Like I said, these are very small changes, so they're looking at different scattering. They had down to a sub-picosecond resolution. What they ended up doing in order to analyze this information was to do a form of single-value decomposition that extracted 4 dominant basis signals. They actually did this single-value decomposition using essentially a model of kinetics. So it's not exactly single-value decomposition, but it's very similar. And so doing that, they were able to actually break this down into 4 time-dependent components, where the first one was some kind of ultra-fast component inside the cofactors that they weren't quite able to resolve even with 500 femtosecond time resolution. C2 was what they had been hypothesized as the protein quake. And you notice that C2 actually precedes the heating components. And so the idea, what they showed here, was that through their structural dynamics with the wax, they were actually able to show how the protein quake essentially rippled through the protein, dissipated that energy before the heating component actually took over. So they're actually able to make a little bit of a movie of that protein quake, that C2 component. And the way they did this is they used molecular dynamics simulations. They had a very high resolution known structure of this protein, which is the photoreactive center from Bacillus viridis. And they were able to actually fit the different frames of that trajectory to the wax profile. Essentially what they do is they take the calculated scattering at some time point in their molecular dynamics trajectory, subtract the time 0 calculated scattering, and fit it to their basis vectors. And they're able to show that they agree very well with their experimental data. So they were able to actually reveal this structural and thermodynamic change at extremely high time resolution. Subsequently, there was also Sachs done at XFELs. This work by Levantino had actually combined Sachs and wax. So one of the potential criticisms of the previous study was that they had put so much light on the thing that essentially was not physiological. Essentially, this is not 1 photon per protein. They were just bombarding it with energy. And so what Levantino did is they did something similar, but with myoglobin, where they actually used a physiological amount of light and used Sachs to actually verify that this protein quake actually takes place. Because the protein quake should not only have high resolution changes, it should actually have low resolution changes because it's the entire structure of the protein that's changing. And so you can see that as a function of time, the Sachs region here changes, and they fitted this with what's called Guinier's law, Guinier's law here, and they were able to get information about the radius of gyration and the volume of the particle. But they also showed that there were high resolution changes as well. Their results showed that they were able to actually see this oscillation in the radius of gyration as a function of time, and they attributed this to this oscillation in the protein quake. Essentially, you have a distribution of mass proceeding from the center. One of the things that was very interesting was that when they looked at the radius of gyration versus the volume, they actually found that the radius of gyration changes slightly faster than the volume change. And so what that suggests is that the mass distributes from the center of the protein, essentially pushes it out, but the volume of the protein stays the same until a brief second later when it actually reaches the edge of the protein, it's able to flex. And so they were able to actually show that that protein quake occurs not only with these large energies that previous experiments had shown, but even with physiological levels of light. Okay, the last thing I'm going to talk about real briefly is something called fluctuation scattering. So one of the main advantages of XFELs is that the pulses are so short that not only do they proceed through the sample before radiation damage takes place, they can actually also outrun any kind of rotational diffusion, okay, which is a much slower process. And so they actually take a snapshot of the protein in one particular position. Even though you may have a solution of proteins, normally in a Sachs or wax experiment, they're tumbling over the course of the experiment and over the course of the exposure. And so what you end up with is actually a sum of single molecule diffraction patterns from random orientations. And you get patterns that potentially will look like this, where there's some fluctuations, but it's not necessarily single molecule, so you can't analyze it directly as single molecule, but it's an anisotropic pattern. This was a method proposed by Cannon back in 1977, and the idea was to use this fluctuation scattering to actually extract information from the correlations in the data rather than directly from the intensities themselves. And so on the right here, he's got some example images where he's got little grains of rice. And you can see that a single grain of rice has a single molecule diffraction pattern corresponding to the top here. Two grains of rice in different orientations, you just sum up their single molecule diffraction patterns. And if you have more and more and more grains of rice, you end up with more and more and more single molecule diffraction patterns, up until you end up with so many molecules that it essentially just becomes a Sachs wax pattern. It becomes isotropic. The advantage of this is that there are orders of magnitude more information content in these fluctuation scattering profiles than there are in Sachs wax profiles. And you can see essentially the way Cam proposed this was to analyze the correlations. So you look at the diffraction patterns and you say, well, what is the relationship between different angles of Q space in our second dimension that we now have, since it's anisotropic? And so this is a plot essentially of that, that correlation profile that he was analyzing. And so the reason he's able to do this is because what it does is he was doing it with physically frozen samples. Essentially, he took something and he froze it nice and then shot it with a beam. An XFEL allows us to do this by temporally freezing the sample. So the particle is frozen in space because it simply just doesn't have time to rotate. And so the idea is that you can do this with XFELs very efficiently. You can have a solution of molecules pass through the beam, and the pulses are short enough to freeze the positions of the molecules over the course of the pulse. And so we call this diffract and destroy, because the pulse actually terminates before the radiation damage takes place, and then the molecules are essentially just ripped apart because there's so much energy. It enables extremely high doses, improving signal noise, comparative to synchrotrons, where you have to actually take into account this damage. It allows you to get to much higher levels of radiation. Okay, so there are several different mechanisms for reconstructing shapes and information, three-dimensional information, from these fluctuation X-ray scattering profiles. But essentially they work based on, they all use spherical harmonics. It's actually quite complicated math, for me at least. And the idea is that you have your diffraction profile as a sum of single molecule diffraction patterns, and you compare the scattering at different angles. And so you have a function essentially at intensity of Q versus Q prime, I guess you could say. And so you end up with a series of different profiles where you have the correlations as a function of Q from the center. You can deconstruct that into a series of essentially basis functions that, using spherical harmonics expansion, you can ultimately reconstruct the electron density function. Now, I oversimplified that greatly, but you can do this. There are different procedures for actually determining the structures. Now, in these cases, you don't have a three-dimensional set of intensities, and that's the big issue. So not only do you have to solve the phase problem, but you have to actually also solve the intensity problem, because you don't know what your amplitudes are in 3 dimensions. So you use these correlations, these cross correlations, in order to determine what the intensities are as a 3-dimensional function. But they do phasing, just like they do in single molecule work. And essentially, they do that by using real space restraints. Things like, I know my molecule has only a finite support. In other words, it's got some limited extent to it. It's not an infinite size. And so we can essentially flatten the solvent. We know that electron density is positive. And in many cases, particles have symmetry. So we can apply all these things as additional restraints and solve the phase problem. So compared to single particle imaging, there's some disadvantages. One is, the primary one is obviously that rather than getting this 3-dimensional set of amplitudes, you have this indirect derivation of these amplitudes, and so there can be errors introduced there. There's a much higher background in solution in liquid than in aerosol, which is aerosol is typically what you use for single molecule imaging. Despite what you may think, just sort of intuitively, is that having more particles in the pattern does not actually increase your signal to noise. And so I guess that's not technically a disadvantage. It's just not an advantage that you might necessarily expect. But there are several advantages. Primarily, they're just practical advantages. There's a lot more information in single molecule imaging, but single molecule imaging is a lot more difficult to do. One of the major advantages is that inherent to the method is that you can use multiple particles per shot. With single molecule imaging, you need one particle per shot, which is very difficult to do. It enables solution-based injection versus aerosol. Essentially, because of this idea that you can get multiple particles per shot, you have a 100% hit rate. So every image has useful information. And very importantly, it's a relatively straightforward merging of the data, whereas with single molecule imaging, you essentially have to store hundreds of terabytes of data and then analyze each one of those to get information about their orientation and correlations for orientation determination, and then to merge all the intensities in 3 dimensions. Whereas with fluctuation scattering, you just need to know the correlations. So essentially, you can analyze the correlations of each image and then just sum them up. The correlations of each image on top of the other images converge to the correlation function of a single image, and that's the basis of the idea. So there's a lot of advantages. And just a real quick slide this is some simulations that have been done that show that you can use fluctuation scattering to get very high resolution information. In this case, 4.8 angstrom compared to Sachs or wax, which allows you to get essentially 0 ab initio shape information out to this resolution. And you can reconstruct with these symmetry restraints, in this case, very close models to your original electron density function. So real quick, XFELs obviously enable radiation damage-free solution scattering. It's a huge advantage over synchrotrons. With XFEL Sachs, you can monitor time resolved changes in things like the conformational state and the size of the molecule, whereas wax can give you information about small structural changes if you have known information, and can do MV simulations. And one of the very exciting things about XFELs is that they provide an entirely new way of doing solution scattering called fluctuation scattering, which greatly increases the information content compared to other solution scattering methods. So that's it for me. If there's any questions, happy to take them.